Bismillahirrahmanirrahim. Assalamu alaikum warahmatullahi wabarakatuh. My dear subscribers, I hope that you are all doing well and great in your lives. Welcome back to my YouTube channel Learn Biology with Nas with another interesting video that is about organelles. Organelles are actually small structures within cells that perform dedicated functions. These are the assemblage of biomolecules, and there are about a dozen types of organelles commonly found in eukaryotic cell. Following are some of the major organelles found within eukaryotic cell. Let us discuss each of these in detail. Nucleus. Nucleus is the organelle of eukaryotic cell where genetic information is stored. A prominent nucleus occurs in eukaryotic cell. In animal cell, it is present in the center, while in mature plant cell, due to formation of large central vacuole, this nucleus is pushed to a side. Nucleus has following parts: nuclear envelope, nucleoplasm, and nucleolus. As for there is the matter of nuclear envelope, so nucleus is bounded by a double membrane known as nuclear envelope. This envelope contains many small pores that functions to semi-permeable membrane. On the second number, there is nucleoplasm. Inside nuclear envelope, a granular fluid nucleoplasm is present. It contains one or two nuclei and chromosome. Nucleolus. Nucleolus is the dark spot, and it is the site where ribosomal RNA are formed. Chromosomes are visible during only cell divisions, while during interphase they are found in the form of thread-like structure, and those thread-like structures are termed as chromatin. On the second number, there is the organelle that is ribosome. It is also called protein factory of the cell. These are tiny glandular structures that are either floating freely in cytoplasm or are bounded to the endoplasmic reticulum, that is the organelle. As for there is the concern of discovery, in 1955, George E. Plade discovered ribosome. Ribosomes are not bounded by membranes and so are also found in eukaryotes. Eukaryote ribosomes are slightly larger than prokaryotic ones. Composition of ribosome. A ribosome is made out of RNA and proteins, and each ribosome consists of two separate RNA protein complexes known as the smaller and larger subunits. The larger subunit sits on the top of the smaller subunit with an RNA template sandwiched between the two. Ribosomes are the site of protein synthesis, and protein synthesis is extremely important to the cell, and so large number of ribosomes are found throughout the cells. When ribosome is not working, it disassembles into two smaller units. The next organelle under discussion is mitochondria. A double membrane bounded structure found in eukaryotes is known as mitochondria. These were first discovered by 1857 by psychologist Albert von Kolliker and later coined bioblasts by Richard Altman 1886. Bioblast means life germs. If we talk about the structure of mitochondria, so the outer membrane of mitochondrion is smooth, but the inner layer forms many enfoldings, and those enfoldings are called cristae. Mitochondria are the sites of aerobic respiration. These generate most of the chemical energy needed to power the cell's biochemical reaction. On the fourth number, there are plastids. Plastid is another organelle, and these are membrane-bound organelle that occurs only in the cells of plant and photosynthetic protists. There are three types of plastid: chloroplast, chromoplast, leucoplast. Chloroplast. It is bounded by a double membrane. The outer membrane is smooth, while the inner membrane gives rise to many sacs called thylakoids. The sacs of thylakoid are called granum. Grana floats in the inner fluid of chloroplast, and that fluid is called stroma. Chloroplasts are the sites of photosynthesis in eukaryotes. They contain the chlorophyll and associated pigments. These pigments are present in thylakoid of grana. Chromoplast. Chromoplasts contain pigments associated with bright color and are present in the cells of flower petals and fruit. Chromoplast. These give bright color to the flower and fruit, so help in the pollination. These also help in the dispersal of fruit. On the third number, there is a type of plastid that is leucoplast. Leucoplasts are colorless and store starch. Starch proteins and lipids there are present in the cells of those parts where food is stored. The next organelle is endoplasmic reticulum. It is the network of interconnected channels that extend from cell membrane to the nuclear envelope. These were discovered by Emilio Viretti in the year of 1902. On the basis of their appearance, there are two types of endoplasmic reticulum. The first one is rough endoplasmic reticulum, while the second one is smooth endoplasmic reticulum. Rough endoplasmic reticulum is so named because of its rough appearance due to the numerous ribosomes attached to it. Function. As many ribosomes are attached to RER, so it serves as in function of protein synthesis. Smooth endoplasmic reticulum. Its surface is smooth because it don't contain ribosomes. That's why it's named so. It is involved in lipid metabolism and transport of material from one part of the cell to the other. Smooth endoplasmic reticulum also detoxifies the harmful chemical that has entered cell. The next organelle under discussion is Golgi apparatus. Golgi apparatus is the set of flattened sacs, and those sacs are termed as cisternae. In this set, many cisternae are stacked over each other. The complete set of cisternae is called Golgi apparatus or Golgi complex. Golgi apparatus were discovered by an Italian physician called Camillo Golgi. Golgi apparatus modifies molecules coming from rough endoplasmic reticulum and packs them into small membrane-bounded sacs called Golgi vesicles. These sacs can be transported to various locations in the cell or to the exterior in the form of secretions. Lysosomes. Lysosome is the next organelle, and these are membrane-bounded organelles in the cytoplasm of eukaryotic cell containing digestive enzymes enclosed in a membrane to break down the targeted material. That target material can be food or some waste material. In the mid-20th century, a Belgian scientist, Christian René de Derve, discovered lysosomes. Lysosome contains strong digestive enzyme and so work for the breaking down of food and waste material within the cell. During this function, a lysosome fuses with the vacuole that contains the targeted material and its enzyme break down the material. Centrioles. It is the next organelle found in eukaryotic cells. Animals and many unicellular organisms have hollow and cylindrical organelle known as centriole. Each centriole is made of 9 triplets of microtubule, and microtubule are made up of tubulin protein. Centrosomes and centrioles were discovered by Theodor Bowen. Now, what are centrosomes? Animal cells have two centrioles located near the exterior surface of nucleus. The two centrioles are collectively called centrosome. The function of centriole is to help the formation of spindle fiber during cell division. In some cells, centrioles are involved in the formation of cilia and flagella. The last organelle of today's topic is vacuole. Cells have many small vacuoles in their cytoplasm. However, when a plant cell matures, its small vacuole absorb water and fuse to form a single large vacuole in center. Cell in this state become turgid. Vacuole were first discovered by a Dutch scientist Antonio van Leeuwenhoek. Cells have many small vacuoles in their cytoplasm. However, when a plant cell matures, its small vacuole absorb. Sorry for the repetition. Many cells take in material from outside in the form of food vacuole and then digest the material with the help of lysosome. So there is another type of vacuole that is called contractile vacuole. Some unicellular organisms uses contractile vacuole for the elimination of waste material from their body. So far in this video, inshallah, we will meet in the next video with the new topic. Till then, take care of yourself and your beloved ones.